inviting me to speak. I was actually invited by my technician who worked with me when I was in grad school. And so I'm going to be talking about some of the work I did in grad school and then some of the follow-up monitoring and work that I've been doing since working for the BLM. So I'm Rachel Burke. I'm a biologist in Las Cruces, and my talk is titled Mapping Nectarivorous Bat Habitat from the Nectary Up and Implications for Agave Conservation in the southwestern US. So I'm just going to give a brief intro about nectar feeding bats and some of their food plants, and then talk about some of the migration mapping I did for our migratory species of nectar feeding bats, and then some of the local resource availability mapping I did, which was looking at agave density and nectar production to try to identify nectar hotspots in southern New Mexico. And then I'll just talk a little bit about some ongoing work, stuff we have going on with research partners, and also potential climate change implications and indirect impacts to agave. So we have 3 species of migratory nectar-feeding bats that reach their northernmost extent in the southwestern US. So these are the Mexican long-tongued bat, Choeronycteris mexicana, the Mexican long-nosed bat, Leptonycteris nivalis, and the lesser long-nosed bat, Leptonycteris yerbabuenae. And it's their long noses that allow them to kind of get into big flowers and get nectar and incidentally pollinate them as well. And it's these 2 species of Leptonycteris bats that are more of conservation concern. Leptonycteris nivalis is federally endangered in the US and Mexico, and Leptonycteris yerbabuenae was recently delisted in both countries, and we're currently in the US in the 5-year recovery monitoring period to make sure that they're still doing okay, even though they've been taken off the endangered species list. So these bats are the primary pollinators of many species of agave and columnar cacti. I believe it's something like 60% of agave or of columnar cacti in Mexico are reliant on bat pollination. They're important seed dispersers, so they also consume columnar cacti fruit. And when they do that, they'll kind of take the fruit to a tree or somewhere where they can rest while they're eating it, and incidentally will drop the seeds or poop out the seeds under a tree, which is really important for providing nurse plants for those younger succulents. And they fly really far between their food patches, up to 50 kilometers in one night. And that's really important for reducing genetic isolation in their food resources because they're transporting pollen from isolated populations of agave or columnar cacti really far distances. And the southernmost populations of these bats have year-round food availability and are typically non-migratory. But their northern counterparts follow what people call a nectar corridor of columnar cacti and agave species. So they follow the plant phenology of available nectar across their migration corridor. And for those of you who haven't seen a nectar feeding bat before, here are a few photos. I believe this is the lesser long-nosed bat pollinating agave palmeri, it looks like. And then saguaro cactus and more agave. So I looked at habitat for all 3 of these migratory nectar feeding bats for grad school, but for the sake of time, I'm just going to talk about the lesser long-nosed bat. And that's also more of a Arizona issue because the Mexican long-nosed bat could technically get into like extreme southeastern Arizona, but it's not really thought to occur in Arizona. So long-nosed bat migration follows the phenology of available food resources. kind of along the Sierra Madres. And previous studies have looked at this and made inferences using indirect methods. People have done things like look at stable isotopes, which is essentially predicting what species these bats were feeding on based on photosynthetic pathways. So like columnar cacti and agave species have a CAM pathway, whereas their more southern plants, the more tropical plants that they feed on are typically C3 plants. Those are the more like tropical trees that these bats will also feed on. But I wanted to know if we could use GIS and spatial modeling to map and predict migration routes and look at the impact of their food resources on their distribution that way. So to do this, I decided I was going to use presence-only species distribution modeling. And this allowed me to take advantage of occurrence records of these bats all over their range. They didn't have to be randomly collected or anything. The software kind of deals with a lot of those biases for you. And then where to start, I used museum records of these bats to do this. They're federally endangered and they were at the time I was doing my project, and have a huge range. So I couldn't just go collect a bunch of occurrence records myself. So I relied on studies and collections that people had already done. And Leptonycteris yerbabuenae, so the lesser long-nosed bat, has been consistently confused with the Mexican long-nosed bat for decades. And a few decades ago, the genus was revised. So a lot of historic records are misidentified. So to really map the distribution of these species separately, I needed to verify these historic records. So basically, I sat in a dark room with a bunch of dead bats and measured specimens for one full summer, maybe more. This photo is actually from the mammal collections at the University of Arizona. And so here you can see a lesser long-nosed bat on the left, the smaller guy, and a Mexican long-nosed bat on the right. And the Mexican long-nosed bat is just like a little bit bigger of a bat. It's a little more robust, and it's hard to tell in specimens like this, but it's got a little bit more fuzz on the membrane between its back legs here. But it's this feature here, the last digit of the third phalange, that differentiates the Mexican long-nosed bat from the lesser long-nosed bat. It's a few millimeters longer on the Mexican long-nosed bat. And I will have cuter photos to show you in a little while. I didn't mean to only show you guys a bunch of dead bat specimens. But they were really beneficial in getting all those records to make all these maps. And because I wanted to look at the impact of food plant distribution on the distribution of these bats, I also wanted to make a bunch of maps of their food plants. So to do this, I gathered all the records I could find of their documented food plants. So I mainly used SciNet, but also used GBIF, and I believe that pulls records from things like iNaturalist too. And I integrated that with phenology information to determine the seasonal availability of these food plants. So if the plant was flowering when the herbarium specimen was collected, I would take the date on the tag of that herbarium specimen and say, okay, this species is known to flower during this month. And then I classified these food plants based on their season of availability. So for that, I used all these herbarium records, but I also used documentation in the literature, and then basically just tallied up how many records or citations I had of these plants flowering in a particular month, and lumped them as being winter, spring, summer, or fall food plant, or multiple seasons. So then I took these food plant records and integrated them into distribution models. It totaled 53 plant species, so it's mostly agave plants and columnar cacti, but it also included some tropical trees and vines that these bats also feed on. So I fed these into the distribution software, and it spits out a map that is the probability of occurrence for that particular species. Then I stacked those and made species richness models. So this map here is a species richness model of all plants known to be consumed or documented to be consumed by the lesser long-nosed bat. The warmer the color, the higher the richness. So you can see there's really high species richness in South Central and Southern Mexico. And that just happens to be where these bats don't migrate because they have food availability year round. So then I wanted to take all of those food plant models and map the seasonal distribution of long-nosed bats and see if we could use those to determine if the lesser long-nosed bat really, if these maps do reveal a corridor of available food resources. And then from those distribution models, we derived migration corridor models. And this was to kind of help identify where there might be stopover habitat. So for these bats, we know where there's really important roosts. We know where there are maternity roosts, and these are well documented and surveyed every year, especially as part of the recovery monitoring process. But we don't know where their stopover habitat is and where they're just staying for really short periods of time during migration. And so with all of these models, we found that food plants were consistently more important than climatic variables at determining the distribution of these bats, and that species richness was the most important factor in looking at the distribution of the lesser long-nosed bat and also of the other species we looked at too. So just here's just a really not that friendly to look at graphic of what that means. But so on the x-axis, we have species richness, and this is for all seasons, and the probability of occurrence for this bat. So you can see that like if we have a predicted 15 species present, you have a greater than 60% likelihood of this bat occurring in that region. And so here are the distribution models for the lesser long-nosed bat that we derived from all of this modeling. And so on the top left, we have winter, and top right, spring, bottom left, summer, and bottom right is fall. And we found that winter was driven by climate, which isn't that surprising because these bats aren't really thought to be that cold tolerant. So they're going to be going to warmer areas in the winter. We found spring was driven by Pachyserius pecten aboriginum, which is a columnar cacti, and these bats will use the fruit or flowers of that cacti, species richness of food plants, and then also precipitation. And then summer, so down here on the bottom left, was driven by species richness of food plants, Stenocerius thuberi, which is another columnar cacti, Crescentia alata, which is more of a tropical tree, and that probably explains some of this distribution down here in South Central Mexico. Carnegie gigantea, so the saguaro cacti, and Agave palmeri, and those are all distributed throughout this Madrean archipelago region where you've got these really red high likelihoods of occurrence in the Sky Islands. And pretty similar for the fall, the distribution was really similar and is driven by species richness of food plants, Agave palmeri, Stenocerius stellatus, which is another columnar cacti, precipitation, and then Agave collimana, which does not occur in the US, but is more of a Mexican agave. So then we fed these all into migration models. We use a software that takes like a suitability surface and can map the connectivity of known sites. So the really red colors here show really strong connectivity between sites. And so here we have winter to spring, and then spring to summer. You get a lot of really strong connectivity starting to happen in southern Arizona into New Mexico. Same goes with summer to fall. And then fall to winter is when they start to go back into south central Mexico. And it was really exciting getting these results because these are basically just the lines on a map that people have just inferred is a migration route, but we kind of were able to spatially prove that that is like the most likely migration route. But then I got really sick of just sitting at my computer and doing GIS all the time. So we're going to go to the field. And because a lot of my work was funded by New Mexico State University and the Las Cruces District Office for the Bureau of Land Management, my work focused on southwestern New Mexico. So for these bats, resource selection at a finer scale, so when you're getting down into these seasonal habitats, not just at the continental, really coarse scale, it's ultimately going to be driven by seasonal food plant density. So not just the presence of their food plants, but the density of that resource availability, as well as plant phenology. They obviously can't feed on a plant that's not flowering or fruiting. And then also nectar production. And Agave palmeri is really the main plant for these bats in southwestern New Mexico. So that was the main focus here. So one of the goals I had with this part of the project was to be able to map nectar availability in southwestern New Mexico. And so to do this, I conducted density surveys of Agave palmeri, and I integrated that with historic data to map plant density. And when I started, I did this all over southern New Mexico and southern Arizona, and we were doing random plots stratified by habitat suitability for Palmer's agave, and doing belt transects within those plots. So this is my tech Chelsea doing these belt transects. But then we kind of found these were taking a really, really long time, and we wouldn't really get that much different information than if we just went to a plot center and kind of used a rangefinder and paced out the plot to just get a count of how many individual plants were within that plot. So that's what I wound up doing to increase my sample size down the road. And so these are all the points that I wound up feeding into my model. And so this is a lot of plots that I read myself, but also some historic monitoring data from the BLM, and then some photo point data from our district botanist, Patrick Alexander, who has basically traversed and took botany surveys at, I would say, the majority of southern New Mexico. And so I took those points and I fed them into a regression analysis with a lot of landscape variables like slope, aspect, elevation, and then also soil and geology. And that was really important to do because at those really coarse models we had, we weren't able to get down to that level of detail. We actually couldn't include soil at that continental scale because that data just wasn't available. But obviously, soil is one of the biggest drivers of plant distribution. So I think that we started to get a lot more accuracy when we got down to be able to include those variables. So here we have a map that's the predicted density of Agave palmeri, and that's within a 1 hectare. So like each pixel on this map is 1 hectare and represents how many agave are expected to occur within that. So the really warm colors have much higher density, and you can see we have these agave hotspots in, these are the Animas Mountains in southwestern New Mexico in the Boot Heel, and then the southern Palancios also. And I wasn't able to include Arizona in this analysis just because it's like so fine scale, but something really similar could pretty easily be done in Arizona too. And then to integrate this with nectar availability, we conducted nectar production surveys in areas with high densities of agave. And that was just to increase the sample size we were able to get at each site because it's pretty intensive to get these samples. And we used a handheld refractometer and tuberculin syringes to monitor caloric output at various floral stages. So we followed methods by Liz Lawson, who's a botanist who's worked in Arizona and New Mexico and has done a lot of agave work before. So what we did is we would identify the sites we were going to survey, and we would go out right before dark and bag our flowers and then come back at sunrise and take our samples. And that's because agave plants produce their nectar overnight, and we wanted to bag them so that other species or even the bats themselves weren't collecting the nectar that we then wanted to sample the production of. So we focused on a variety of floral stages. And so this branch of agave, we would classify this, well, we'd probably divide it in half, but mostly classify it as an early pistillate stage. So you can see the anthers are out, but also the pistils are just starting to emerge. So we would identify that, and then we would bag it and take our samples at like 4 or 5 a.m. the next day. And I'm just going to show you guys some plant photos of what all went on in this. Okay, so what did we find with this? We found that agave produces very high amounts of caloric availability. And this is corroborated by some other papers that have done really similar work. And there was quite a bit of variability amongst floral stages. So we looked at dehiscent, which is like just when the bud is starting to open and the anthers are just starting to peek out. Post-dehiscent, you'd have your anthers all the way out, but you wouldn't be able to see the pistil of the flower yet. Then early pistillate would be that photo I showed you where the pistil of the flower is just starting to emerge. Pistillate would be the pistil is basically level with the anthers. And then late pistillate, everything would kind of be starting to dry up and wilt. And so it was this early pistillate phase that produced the highest amount of calories. So 34 about calories in one night in one individual flower. And so we did that by looking at the production, the total production of the nectar, and also the sugar concentration. And there's just an example of what that looks like. And so I really wanted to be able to map nectar hotspots with this work. As I got more into it, I realized that would take an excessively high amount of data that I did not have. But to kind of just show you what this would entail, say we just made an assumption that within our agave patches, 10% were in bloom, each agave had 20 branches, and 4 were post-dehiscent, 4 were early pistillate, and 4 were pistillate. The rest are now open, and each branch has 50 flowers, which that's actually probably an underestimate. So that would produce a caloric availability model like this. And so again, you're getting really high caloric availability in these nectar hotspots. And if I clicked on one of these really red pixels in the Animas Mountains, for example, in 1 hectare, there are over 130,000 calories available in one night. And that's only looking at one species of agave. So in the future, this could probably integrated with phenology models to make dynamic caloric maps, which I think would be really helpful when trying to predict migration for these species at a finer scale than I was able to with those other models. So we found there's extremely high energetic availability in areas with high agave density. These are mainly in the southern Palencios and the Animas Mountains. And we always talk about blooming agaves for bats, but they also support an entire ecosystem of pollinators from hummingbirds, carpenter bees, bumblebees, and then bats, and tons of things in between. So they're just a really important plant on the landscape in general. And then if we do integrate these density models And nectar mapping with phenology data, this could help guide management and future monitoring efforts, help us identify where stopover habitat is. And our research partners are working on phenology models that can help us better understand this variability across the landscape, and especially with changing climate. And then related to that, disturbance and indirect impacts, both from management and climate change, can impact agave density and nectar availability in areas that are otherwise suitable habitat. So like what I mapped here, you can kind of think of as nectar production potential. But if there's things on the landscape that disrupt that, that site might not live up to that potential. And so some reasons for that, there's researchers looking at range contractions in agave from climate change and what that might mean for plant pollinator complex. Then we also have things like buffalo grass and layman's love grass, invasive grasses in the southwestern United States that alter fire regimes. And so Palmer's agave might be slightly adapted to fire, might not be totally killed off by fire and can do okay with low intensity fires. But these higher intensity fires that we get from these invasive grasses are probably too much for these species. And then we also get herbivory of agave stalks by cattle, but also by native ungulates. And I think, I mean, I haven't done surveys to validate this, but once we have these droughts like we're in now, and there's very little perennial grass availability on the landscape, that's probably going to put increased pressure on agave because those stalks are one of the only things on the landscape that has moisture for ungulates to consume. So yeah, there is a ton of potential for nectar production in these areas, but we need to make sure that we're managing to allow that nectar to become available. And so I just want to thank my thesis committee that worked really closely with me on all of this, the BLM, all my field help, and then funding sources. And with that, I will take any questions.
Hey, Rachel, I have a question.
Hey, Mallory.
Great talk. It was super interesting. I like the pictures. So I'm just continuing the phenology surveys in this area, but I haven't been doing like the different stages because I didn't think to do that, I guess. But would the data of just open fresh flowers be useful at all for the caloric intake or caloric availability throughout the landscape?
I think it would be, and I think it's probably a balance, like because these types of, so I did some phenology surveys where I was looking at that level of detail with the floral stage, and it takes a really long time to do. So it's like just looking at open flowers, I think you could kind of model an average all of those different nectar production values for all the different floral stages, because yeah, I think you would have a really hard time getting a big enough sample size to say anything meaningful if you were looking at that level of detail across the whole landscape.
That makes sense. And I've been doing like 1% of the plant, but most of our plants have about 20 branches. But would it be helpful if when we went around, we just counted how many branches each plant have and like collect that data as well so we could get the branching?
Probably.
Okay. Yeah, I'll probably add that on next week. Just get that data just in case. Cool.
Yeah,
great job.
Thank you.
Other questions or comments? Barb, are you there?
Yeah, can you hear me?
Yes.
Okay. Sorry, I think I was getting kicked off for a second.
Uh-oh. Did we lose Rachel?
Oh,
Is there?
I'm
Yeah.
here.
Okay.
Sorry, technical issues.
Oh, yeah. I just had a question about virus in bats, and will it be affecting agaves? Or do they have enough pollinators, even if something occurred in the bat populations,
Mm-hmm.
they could just utilize other pollinators? I mean, you're not seeing in the Mexican bats that white nose virus?
Yeah, so the white nose syndrome affects bats that hibernate. That's when it kills the most bats. It'll essentially irritate them. They'll wake up out of hibernation, and then in colder climates, there's not food to eat. So they essentially starve from it. They're not worried too much about the effects that white nose might have on long-nosed bats because they migrate and they go to warmer climates. But there is concern that they could spread it to other species because they travel so far.
Okay.
But yeah, fortunately, we have detected it close-ish to their range, but nothing too close yet.
Okay.
Um,
You're not seeing agave populations being decreased by decreased pollinators in southern New Mexico?
think so. In Mexico, a lot of agave relies like explicitly on bats,
Mm.
but because we're kind of at the northern extent of the range for these bat species, it's thought that there's so much natural variability in the bat populations that the agave have adapted to not completely rely on bats in this area. So they'll also use carpenter bees are a really big pollinator
Okay,
for them, bumblebees, all sorts of things.
good. Good. I'd hate to see a decrease in agave
Mm-hmm.
because of it. They're beautiful.
Yeah. I think invasive grasses are probably like one of the biggest threats.
And as far as the agaves that, for example, are in Verde Valley, way north, they're being pollinated by other pollinators and possibly northern Arizona bats.
Mm-hmm. Yeah, there's actually some new papers that pallid bats, which are an insectivorous bat, actually pollinate agave too.
Okay.
But yeah, there's all sorts of insects that'll pollinate it.
I see bats in the evening, and we have no real agaves here. It's cold. I
Yeah.
can't begin to grow them. I have so many trees.
Yeah.
I think Patty gets to grow agaves.
Well, the last couple of years, we've had the fine leaf yuccas coming up all on their own and blooming. And I'm thinking we're becoming the desert up here. I mean, we live a little bit more distant from the mountain and we don't get as much rain. We don't get as much snow. So I think we're on the verge of being the desert. So I planted an agave and it just did beautifully. So yeah.
Yeah, some of them are pretty cold hardy. They're probably just a little harder to get established when it's that cold.
Yeah, and when I bought agave for and cacti for our garden, I was really careful to get some that were cold hardy, but those yuccas just came up on their own and I didn't invite them, so. But they're nice, they're pretty and it beats cheatgrass or whatever.
Yeah.
Yeah.
So what do you think is the answer to the grass problem? I mean, you have a strategy or a theory about what to do about that?
I think
Because it's a threat to everything.
Yeah, it's complicated, and a lot of them were planted like back in the 50s when we were still more about planting non-native grasses for forage and erosion control. I know in Arizona, they do a lot of volunteer work for buffalo grass removal. For layman's love grass, it's a little bit more naturalized in Arizona at this point. It might not be as big of an issue as buffalo grass in terms of like fire intensity. But I think a lot of the issue is these grasses will spread and establish once we allow the native grasses to get degraded. So I think preventing that from happening and trying to keep our native grasses as healthy as we can is probably pretty important.
We don't really have a big buffalo grass issue up here.
Down south, yes. Tucson has a horrible
Yeah.
situation with it. Yeah.
I still think the big thing here is cheatgrass, and and there's, you know, you can handle it in your yard, but it's just everywhere.
Yeah. Fortunately, cheatgrass isn't really an issue in the range of palmers agave.
Oh, okay.
Okay. All right. This was very interesting. Thank you so much.
You're welcome.
Really appreciate your talk.
Yeah, thanks for inviting me. It was fun.
It's fun because we can be in totally different places and still hear all good news about research.